Welcome, everyone, and thank you for tuning in. Today, we are talking about cancer as a metabolic disease. Joining us in today's conversation is Professor Thomas Seyfried. Professor Seyfried is a leading researcher in cancer biology and metabolic therapy. He has his doctorate degree in genetics and biochemistry and is a professor of biology at Boston College. He's renowned for his groundbreaking work challenging traditional views on cancer's origin. Professor Seyfried presents a very compelling argument that cancer is primarily a metabolic disorder rather than a genetic one. He has published a very influential book on this topic called Cancer as a Metabolic Disease on the Origin, Management, and Prevention of Cancer. We're honored to have you with us today, and welcome, Professor Seyfried.
Thank you very much. It's a pleasure to be here today with you.
So I want to start by looking at the two ways most people think of cancer. You say that cancer has remained incurable for many people due largely to this general misunderstanding of its origin, its biology, and metabolism. Can you explain for our listeners the two compelling theories behind cancer, the semantic or genetic theory and the metabolic or mitochondrial energy theory?
Yes, thank you. Of course, to differentiate those two origins is what my book was about, and you really need to go into the deep background of the two concepts, how they arose, how they developed, and where they stand today. Well, the first thing we should know is, people always ask me, what is cancer? And cancer, by definition, is cell division out of control, which could happen in a population of cells in any given tissue or organ, where these cells start to grow, dysregulated cell growth. So, what is responsible for maintaining the regulated state of the cell? So, if the disease is cancer, regardless of whether it's a breast tumor, colon tumor, brain tumor, bladder cancer, lung, melanoma, blood cancers, we have any of these cells starting to grow out of control. And that's what we get. And we give a name to the type of cancer based on the cellular origin or tissue origin. He has lung cancer, he has breast cancer, they colon cancer. Their cells grew out of regulation in these different organs. So the question is, what is causing these cells to become growth dysregulated, and more importantly, what's keeping them in a regulated state? And what we have found is that it's energy, the way the cells conduct their daily businesses, the metabolic homeostatic state of the cell. And these cells are doing things in the bladder, the breast, the colon, the brain, the lung, and they perform a differentiated function. Their activities are differentiated to that specific organ and for the activities of those cells in that organ. Now, then all of a sudden, some of these cells start to grow in a dysregulated way, forming what we call a neoplastic transformation. And they form, when they get enough of these cells together, they form a tumor. And if it's in the blood, they could be just a redistribution of the numbers of cells. And you notice that there are more leukocytes higher than before, white blood cells and this kind of thing. But whatever it is, it's cell division out of control. There are two dominant theories right now to explain how these cells lose their regulated growth and become dysregulated. And the most common recognized theory today is the somatic mutation theory, where sporadic somatic mutations in cells of that tissue become mutated, and these mutations are causing the cell to enter a proliferative state and become dysregulated, caused by somatic mutations. And this means mutations in the tissue cell itself. The somatic mutations differ from germline mutations, where we pass the mutation on from one generation to the next. Those are called germline mutations. But the somatic mutation theory posits that the cancers are caused by DNA defects, mutations in the nuclear DNA that lead to a dysregulation of growth. And you find many different kinds of mutations. In fact, tens of thousands and millions of different somatic mutations have now been recognized. Some of them are common to different tumors, and many of them are unique to a particular tumor, leading the National Cancer Institute in the United States to say that cancer is a genetic disease representing more than 100 different kinds of cancers, all involved in having some form of genetic somatic mutation defects in the nucleus of the cell. That pretty much describes what the standard view of cancer is, according to not only the National Cancer Institute, NIH, NCI, National Cancer Institute, but also all the major cancer centers throughout the world, whether it's Germany, Japan, England, China. They all are locked into the same worldview that these cells are dysregulated because of some sort of defect in the DNA, which is supposed to then lead to precision medicine, which is the development of drugs that are supposed to target the consequences of these genetic mutations to see whether or not we can stop the dysregulated growth and kill the tumor cells. So we have immunotherapies, Opdivo, Keytruda, PD-1, PD-L1, CAR-T immunotherapies, which are really even a more complicated way of doing this. And then we have our standards of care, which are surgical removal, radiation therapy, chemo, toxic chemicals. And many of these toxic chemicals are designed to stop DNA replication. So the mutations are thought to lead to a dysregulated replication of DNA or failure to repair damaged DNA or whatever. And therefore, we have a whole group of drugs that are designed to target DNA replication, prevent DNA repair. We have all these different drugs. A cytoskeleton, which is the proteins in the cell, are needed for cell division. So if you can block them, the problem with many of these therapies is they're not specific to the cancer cells, and they can have very off-target effects. So consequently, you have hair loss, you mucositis in your gums, you have a destroyed microbiome, fatigue, diarrhea, nausea, hair loss. Okay, as I said, hair loss, what is the hair? Doesn't have tumor cells in it, yet the hair cells are dying. The gut is destroyed or significantly damaged. And these are the consequences. Now, you have to realize that most scientists and oncologists are not happy having to treat their cancer patients with these toxic procedures, mainly because that's all they have. And the promise of the multi-billion-dollar cancer genome projects were supposed to be identifying very precise targets for these new drugs to come in to say, let's reduce some of the horrible toxicity associated with these more primitive approaches and maybe move the field into a better state because of our knowledge of the gene mutations that are based on the somatic mutation theory of what cancer is. And we know that many of these new drugs immunotherapies based on the somatic mutation theory of cancer do have some therapeutic benefit in a number of people, but they also have the possibility of actually destroying normal tissues and causing a phenomenon called hyperprogressive disease, where the cancer cells actually grow much faster than they would if you didn't do anything to them. So some of these drugs can make it's called hyperprogressive disease as an indirect effect of targeting, off-targeting, or targeting, trying to attempt to target this. So this is where the field is. The majority of cancer people are being treated with either the traditional standards of care, which are toxic poisons, high-dose radiation, or surgical mutilations. And then if that doesn't work, then they might go to some of these more sophisticated immunotherapies to see if they might work. And a lot of these new drugs are based on drug development based on the somatic mutation theory. Let's look at the results from this. Okay, right now we have 1,700 people a day dying in the United States from cancer. That comes to 70 per hour. Okay, none of these drugs have had any significant impact in improving overall survival. They have what we call progression-free survival, which is really remarkable because many of these drugs, when a patient is treated with some of these new drugs, the tumor looks like it disappears. And you say, this really is powerful. But when you look at the overall survival, it's not statistically significantly increased in any meaningful way. You might get an extra 2 or 3 months of poor quality of life based on one of these new drugs. So it's really not dropping the death rate. As a matter of fact, the more and more people die each year, 70 an hour, getting almost 1,700 a day, and more drugs are poured into the society. So there's something wrong. Why are we not making the kinds of progress? Now, we have millions of cancer survivors who have endured these toxic approaches and these toxic treatments, and many of them are so grateful for their survival. But the vast majority, not all, majority have paid a significant price to live that extra time. They have neuroendocrine problems, bone density problems, neuropsychiatric problems, digestive problems as a consequence of having been brutalized by what I call medieval therapies to give them a slightly longer time on the planet. And of course, they're grateful for that. And this is the standard of where we sit today with cancer. It's recognized as a gene-based disorder. We know that there are some germline mutations that can actually be risk factors, like the BRCA1 mutation for breast cancer, P53 for the Li-Fraumeni retinoblastoma, these kinds of mutations. And we now know that all of those mutations, germline mutations, influence energy metabolism in one way or another. So that I gave you a breakdown now of what we call the current view of cancer is it's a genetic disease caused by somatic mutations that lead to dysregulated cell growth. The alternative and correct theory is the mitochondrial metabolic theory of cancer, which was originally defined by Dr. Otto Warburg from Germany back in the early part of the 20th century. He claimed that all cancers, regardless of tissue origin or species, all have a common metabolic problem. They produce energy in the absence of oxygen, and that was linked to various kinds of defects in the mitochondria of the cell. The mitochondria, you need to know cell biology and cell anatomy. We have a nucleus of the cell, and I think most people realize the nucleus contains the DNA, the majority of the DNA in the cell. And then we have this tubular spaghetti-like network called the mitochondrion, which determines the energetic state of the cell. The mitochondria allow the cell to perform its daily activities in a regulated way. So the mitochondria, when a cell needs to divide, the mitochondria energy allows the cell to divide at a regulated pace to replace or renew certain cells in different organs. This is our natural biological turnover of cells. And that regulation of division is controlled by the mitochondria's energy gradients inside that cell. Now, what we're finding, what Warburg found, and what we have also found and what we've elaborated more on, is that the ability of a cell to regulate energy becomes corrupted and dysfunctional in the cancer cell. Rather than using oxygen to perform its local activities, the cancer cell has a loss of that ability gradually over decades, years. The ability of the organelle mitochondria to regulate energy metabolism gradually becomes inefficient and corrupted. But the cell doesn't die. The cell gradually transforms back to ancient forms of energy that existed before oxygen came into the atmosphere 2.5 billion years ago. So we had living organisms on our planet, single cell organisms that got their energy from fermentation and energy without oxygen because there was no oxygen in the atmosphere. It was only after the development of certain bacteria that produced oxygen in their metabolism did we begin to transform into what we call eukaryotes. These are more sophisticated kind of living organisms, leading eventually to multicellular organisms and very highly sophisticated plants and animals that all have a regulated energy metabolism controlled by the organelle mitochondria, which actually were derived from one of these oxygen-consuming kinds of bacteria. So we see a cancer cell now very different from a normal cell that where you and I breathe, the oxygen comes into our bloodstream, and we allow that oxygen to form energy by the process of oxidative phosphorylation, which is energy generation through a very sophisticated, highly efficient energy process in the mitochondria. Now, as the mitochondria, I should say, become corrupted by any number of different provocative agents, insults in a particular population of cells over time, from like, let's say, systemic inflammation, a viral infection, oncogenic virus infections, intermittent hypoxia. You can have carcinogens. People always fear carcinogens. Why? It's a chemical that causes cancer. How does it cause cancer? Well, it disrupts energy metabolism in the mitochondria chronically, forcing the cell into fall back on these ancient pathways. Now, interestingly enough, all of our cells in our body have these ancient pathways. The problem is they're very minimal because the mitochondria can produce so much energy so much more efficiently. But when that organelle becomes gradually corrupted, the cell falls back on these ancient pathways, which now become dominant. In the old days, when I say during the development of organisms, those cells would grow in an uncontrolled way, completely dysregulated in their growth, and would die as soon as the fuels driving this fermentation energy would dissipate. So the cell would die because it no longer has energy. So the cancer cells are similar to those very early stage cells. They no longer are regulated by mitochondrial energy metabolism called oxidative phosphorylation, and they fall back on, I'm sorry to have to say this word is substrate level phosphorylation. I know I don't want to lose your viewers from the biochemistry of what we know, but substrate level phosphorylation is that can form energy without oxygen. Energy without oxygen, which is those ancient pathways. Otto Warburg said that the cancer forms because the efficiency of the mitochondria becomes gradually dissipated and corrupted, allowing the cell to fall back on a fermentation, energy without oxygen. So this was his great accomplishment. And he said all cancers result from this chronic damage to oxidative phosphorylation, leading to a compensatory fermentation driven by substrate level phosphorylation. And interestingly enough, when you look at it this way, so cancer cells are generating energy in the absence of oxygen. If we were to take cyanide, which everybody knows is a poison, it kills you very quick, cyanide shuts down energy through oxidative phosphorylation. If you and I took cyanide, we'd be dead in a minute because we shut down all energy metabolism. Cancer cells can live in cyanide, clearly saying to us they are not using oxygen, oxidative phosphorylation. They're driven by a fermentation metabolism, and you can shut down their ability to use oxygen, and they'll still survive. That told us.
How do they know? How do we know they live in cyanide? We've done this experiment.
Yeah, Otto did it.
Oh,
I did it. A number of other people have done it. Yeah, they grow
okay.
inside, and they can grow in
Wow.
the absence of oxygen. We grow them in hypoxia, and this is one of the things. That's why cancer cells are hypoxic. You have all these cells growing in a hypoxic environment. They're falling back on a fermentation metabolism, just like the cells did before oxygen came into the atmosphere 2.5 billion years ago. So we need to know evolutionary biology, and if we're going to really understand the cancer issue. So now we know. So the question now becomes, where are they getting their what is fueling this fermentation metabolism? Otto Warburg said it was glucose. And not only that, he said the cancer cells all of a sudden start to upregulate and take in massive amounts of glucose and generate energy through a fermentation metabolism, glycolysis. And normally the end of glycolysis, which is pyruvate, would go into the mitochondria and be fully oxidized to water and CO2. So the waste products of normal respiration that you and I are currently doing, and all normal folks, is water and CO2. So every exhale we have carbons, CO2, and a moisture water, and we generate water and CO2 waste products from normal respiration. Cancer cells dumping out lactic acid as a waste product. Now, if we were to exercise aggressively, we see lactic acid build up in our bloodstream because the muscles can't get enough oxygen to do their contraction. And as the result, muscles can ferment for a short period of time, and you see lactic acid build up real quick. Cancer cells are, what Warburg said, the strange thing about cancer is they continue to produce large amounts of lactic acid, even in the presence of 100% oxygen. So then he said, there must be something wrong with the mitochondria efficiency in cancer cells because they continue to ferment in oxygen and they shouldn't be fermenting in oxygen. And the other clue that we and others have found that Warburg did not know about is we found that others, like when you have a cardiac attack, cardiovascular attack, a heart attack basically, your bloodstream fills up with massive amounts of lactic acid and another form of acid called succinic acid. Lactic acid and succinic acid build up in the bloodstream. And that's because if in contrast to cyanide completely blocks oxidative phosphorylation and kills the cell. The heart attack is just stopping us from breathing, but in order to keep our brain cells and our heart cells alive, they immediately switch to this ancient fermentation pathway with the waste products of lactic acid and succinic acid, but that can only be done for a few minutes. And if you don't get oxygen back into your body, you're going to be dead. The cancer cell is throwing out those two waste products all the time, even in the presence of oxygen. It's dumping out lactic acid and succinic acid. So our big breakthrough in cancer metabolism was finding that the amino acid glutamine. Now we have amino acids in our bloodstream, and they're there as metabolites for a lot of different biological reactions. But the most abundant amino acid in our bloodstream is glutamine. Glutamine is very important for the health of our gut, the health of our immune system, our urea cycle, and it's the most abundant amino acid. So when glutamine is fermented and it leads to a buildup of succinic acid, and like I said, when you have a heart attack, you build up lactic acid coming from glucose fermentation and succinic acid coming from glutamine fermentation, which means these two fuels are generating energy, ATP, in the absence of oxygen using these ancient fermentation pathways. So when Otto Warburg always knew about the lactic acid marker and saying, oh, they have to be fermenting. Lactic acid should never be accumulating under oxygen, nor should succinic acid. And we are the ones who showed that succinic acid, this abundant amino acid, is there to keep us alive during a heart attack for a short period of time, besides doing all these other important functions. So then we know that the cancer cell immediately shifts, not immediately, gradually shifts to these fermentation pathways as the energy efficiency of oxidative phosphorylation over time becomes corrupted. So cancer doesn't start one day and the next day you wake up with a giant tumor on your body. What happens, it's a slow progression of damage to oxidative phosphorylation, mitochondrial energy efficiency, and a gradual transition to a compensatory substrate level phosphorylation or fermentation metabolism. And then we now know what's going on. And because the mitochondria control the differentiated state of the cell, when that organelle becomes corrupted and the cell begins to fall back on these ancient pathways, they become dysregulated in their growth. So the dysregulation is the result of the failure of the mitochondria to maintain energy balance, and the cell falls back on its default state, which is proliferation, exactly like the cells did before oxygen came into the atmosphere 2.5 billion years ago. So these cancer cells are doing nothing more than falling back on ancient pathways. And as long as they have an abundance of fermentable fuels, which are glucose and glutamine, in their microenvironment, they are very hard to kill. They will grow as long as they have those fermentable fuels, and they cannot switch to other fuels. They're locked into these two fuels, and therefore they are driven by fermentation. And then to kill them, you pull the plug on their fermentation and transition the whole body to fatty acids and ketones, which the tumor cells can't use. But the normal cells evolved to switch from these fuels to these fatty acids. Tumor cells cannot do this. Thereby, they become compromised, checkmated, and you can kill them relatively easy without toxicity. Now, what I just told you is either resisted, not known by the cancer establishment. They don't either want to know this, or they can't know it, or they choose not to know it.
So I want to
I know it's a
have
lot.
some,
I gave
yeah,
you
no, this is good.
hundreds of thousands of articles in the literature summarized in those few time periods.
Right. So I have some follow-up questions on some of the things you said, like with the BRCA genes and all that, but I want to take one step just to make sure that it's clear to all of our listeners. And so I just want to summarize and make sure that we have this clear. So what is suggested by the metabolic cancer theory is that there is somehow chronic damage made to the cell's ability to produce energy through oxidative phosphorylation, which is how they produce this ATP, the adenosine triphosphate, right, that provides the cell energy. Then the cell compensates by using fermentation to feed itself, which doesn't require oxygen. And it does this by utilizing, you said glucose, and now you have also found that glutamine is also used in this fermentation process. And when the damaged cell is deprived of oxygen, it relies on these two substances, glucose and glutamine, for fermentation and growth, which is how the tumor, which we defined as some uncontrolled growth, survives. Is that correct?
Yes, and one more thing. that corrupted oxidative phosphorylation in
Mm-hmm.
the tumor cell produces what we call reactive oxygen species, ROS. These are radical. Oxygen is still coming in to the tumor cells, but it's not being coupled to ATP synthesis. It's being used to produce ROS, these radical oxygen molecules and hydroxide ions. And guess what? They are carcinogenic and mutagenic. So the mutations that we see, the millions of mutations and chromosomal damage and all the nuclear damage that the field seems to focus on, are all downstream effects of the chronic disruption of the oxidative phosphorylation. So we know that cancer cells are loaded with every type of kind of mutation, hundreds and tens of thousands of these mutations. And they're all different from one cell to the next and from one cancer to the next. So the question is, where are all these mutations coming from? They're coming from the abnormal energy metabolism in the cell, producing these ROS. Here's the crazy stuff. So you say now, wow, now I know where the mutations are coming from, and they're not the cause of cancer. They're the effects of this damaged respiration. So why would the field continue to focus on things that are not relevant to the nature of the problem? And that's where most of the energy comes in. The NCI says, oh, it's a genetic disease caused by mutations. The mutations are an effect of the damaged respiration and energy metabolism. So constantly building drugs and focusing on these mutations will not solve the problem. The problem is an
So,
energy problem.
so I wanna come back to this fermentation, right? You mentioned in your book that there have not been any cancers found that can live without fermentation. Can you explain the importance of that fact? Because I think that helps to make it a little bit more clear.
Yes, well, this is one of the commonalities that we found for all major cancers. Okay, I
Mm,
had to do a deep dive and I said, okay, Otto Warburg based his theory of cancer as a metabolic disease predominantly on the biochemistry. He said cancer cells have reduced oxygen consumption and increased lactic acid production. That's the compensatory fermentation. So, and he found that in all different kinds of cancers. But he never looked at the structure and function of mitochondria in the sense of what they looked like. And he couldn't have done that because we didn't have the electron microscope developed at the time he was doing his research. So I had to go back into the pathology, the literature of cancer pathology, when the electron microscope was first developed. And we looked, and now we could see mitochondria, and we can see the elaborate structure of this organelle. It has these beautiful stripes called cristae, and the cristae, all of this organizational structure of the mitochondria, they contain the lipids and the proteins of the electron transport chain that allow the cell to get energy from oxygen. So when we started to look at mitochondria, we saw, wow, look at all the structural abnormalities inside these organelles. They had no cristae. A lot of them were ghost mitochondria. And then when we isolated mitochondria in pure form and did enzymatic studies on certain aspects of mitochondrial function, invariably they were abnormal. So we were able to link Otto Warburg's pure biochemistry to visible structural abnormalities. In biology, structure determines function. And if the structure of the organelle is abnormal, the function of that organelle is going to be abnormal. So we now have anatomical evidence saying that the structure and function of the very mitochondria are abnormal, which means that in order for the cell to survive, it must switch to a fermentation metabolism. So we
right.
could actually see that and support that further. So, and when you said the fermentation, okay, what does that mean? That means glucose comes into the cell and is metabolized to pyruvate through the 10-step Embden-Meyerhoff-Parnes pathway called the glycolytic pathway. And then that pyruvate would normally go into the mitochondria and be fully oxidized to CO2 and water, but instead it's being diverted it's being reduced to lactic acid. And that was the signal. The cancer cell is telling us that I'm not able to use oxygen effectively because I'm throwing out all this lactic acid. That's the signal that the cancer cell is not using effectively oxidative phosphorylation, falling back on fermentation using the cytoplasm. Now, what the great mystery was, but cancer cells are still taking in oxygen and still making ATP coming out of the mitochondria. How do you explain that? And when we discovered that the mitochondria, there's a substrate level fermentation mechanism inside the mitochondria. The mitochondria can switch to a fermentation inside the very organelle that's supposed to do oxidative phosphorylation. So glutamine is metabolized to glutamate, to alpha ketoglutarate. I had to go through the TCA cycle. And they dump out succinic acid. Succinic acid should never be dumped out of the cell. That means they're using glutamine and getting energy through mitochondrial substrate level phosphorylation. Now, I had this idea, and I was not an expert on that part of the pathway. I just guessed. They call it a speculation. I said, I think mitochondria are fermenting through this mitochondrial substrate level phosphorylation. And I put together a little diagram, which wasn't completely correct. So I reached out to the world leader, Dr. Christos Xenopoulos from Semmelweis University in Budapest, Hungary. He is the world leader on that particular pathway and that particular mechanism. And I asked Christos, can you see if my speculation might be correct in thinking that the glutamine could be fermented rather than respired, because this is complete mystery to the entire field. So he and I worked together, and we designed, in our big paper that just came out in ASN Neuro, provides the first hard evidence that these damn brain cancer cells are fermenting glutamine. Warburg never knew about this. Warburg did not know this. And as a matter of fact, almost nobody in the cancer field knows about it either. But it explains so much. It explains what we see. The cancer cells have been telling us this for decades. We're fermenting, and the waste products of fermentation are lactic acid and succinic acid. And we're seeing that. Cancer cells continue to produce these fermentation waste products even in the presence of oxygen. So then we tested the hypothesis, well, if the cancer cells need glucose and they need the glutamine to provide the energy for ATP, what happens when we simultaneously target those two fuels while transitioning the body off to a fatty acid ketone? Because you need a good mitochondria to get energy from fatty acids and ketones. Mitochondria defective. You can see it. So the normal cells are able to use ketones and fatty acids, but the cancer cells can't. Checkmate. Right?
So I want to ask you, yes, I see. I'm now going to get into the therapies in a little bit, because I want to go back to something else you said. So couldn't we potentially use those two, the lactic acid as markers to see if the cancer is there still, even when you mentioned there are times when the chemotherapy has its effect and then you can't see it, which is true. You can't see it on the MRI, you can't see it, right? But you're saying it's still there. We just can't see it in the exams.
Yeah, so you're right. I mean that's some blood cancers will produce so much they'll get lactic acidosis from the blood cancers throwing out so much lactic acid, but they also throw out succinic acid. The level of succinic acid is never as high as lactic acid, because the part of that pathway is filtered to use other amino acids and things like this. So, but lactic acid, these are markers of fermentation metabolism. And then we were tracking down, everybody knew for centuries since Warburg's time, 100 years ago, that the lactic acid was the result of a cytoplasmic, in the cytoplasm, fermentation. But the succinic acid, we have this fermentation in the mitochondria itself, the very organelle that's supposed to be so energy efficient. So now we know that actually ferments too. And because it came from a cell that was originally a fermentative cell. And the waste product is succinic acid. And glutamine, glutamine has two nitrogens. And in order to make new DNA and RNA, you need nitrogen. And the glutamine is the part of the fermentation pathway giving us the nitrogen to make new DNA and RNA and other amino acids, where the carbons from glucose are the metabolites for lipid synthesis, also nucleic acid synthesis. So the carbons of glucose and glutamine and the nitrogen of glutamine together work in a powerful synergy to drive the dysregulated growth of these cells, because the organelle that's supposed to regulate this is dysfunctional. So
I wanted to ask you, so what are some factors that lead to the mitochondrial dysfunction in healthy cells, potentially initiating this cancerous process?
Yeah, well, this was referred to as the oncogenic paradox by the late Albert Szent-Györgyi, a Nobel Prize winner from Hungary as well. Albert Szent-Györgyi said, wow, almost everything in the environment can be linked one way or another to cancer. You're exposed to chemical carcinogens, you're exposed to radiation, intermittent hypoxia, oncogenic viruses, systemic inflammation. Any one of these conditions can damage the number and structure and function of mitochondria in a particular population of cells in a particular organ or tissue. So, you get breast cancer. I'm giving you that as an example. A milk
Yeah.
duct gets occluded, leading to a focal chronic inflammation, which damages over time the ability of the cells around that occluded milk duct, causing it to gradually transition to a fermentation metabolism driven by glucose and glutamine. You can have that in a colon cell of the crypt. You can have that in a brain astrocyte in the brain. You can have that in a lung cancer cell. You can have it in a kidney cancer, bladder cancer, melanomas. Every cancer that we have looked at is driven by a fermentation metabolism. How that happens could be any number of chronic exposures to the cells in that particular tissue, leading to this transition from respiration to fermentation. And the reactive oxygen species produced cause the mutations in the DNA. So you see DNA mutations in all these different cancers, but they're arising from damage to the mitochondria coming from any number of provocative agents. So somebody may get cancer from systemic inflammation. Now we know that obesity is beginning to replace smoking as a number one risk factor for cancer. But many obese people have high C-reactive protein in the bloodstream. A lot of them have systemic inflammation, type 2 diabetes, hypertension. All of these kinds of things create stress in the body, throwing us out of metabolic homeostasis, leading to chronic damage to mitochondria in a particular cell or population of cells in a particular organ, leading to dysregulated cell growth and cancer. So driven by fermentation metabolism.
So I want to go back and ask you, because you mentioned this. So for example, for the breast and melanoma, the BRCA1 and 2 genes and the TP53, where the mutation in these genes is very commonly found in these types of cancers. So I guess the explanation from the gene mutation theory would be that those are downstream effects. Is that what you're saying? And what we really need.
Yeah, well, we have two things with P53. We have a germline inherited P53 that damages the synthesis of cytochrome C, which is a protein in the mitochondria. Okay, so clearly the inherited mutation disrupts energy efficiency in the mitochondria of the cells that express the mutation. And the BRCA1 does the same thing. It disrupts oxidative phosphorylation in the cell, but it's a germline. It's inherited through the germline. But the interesting thing, and we've just discussed this, none of these cancer genome germline mutations are 100% penetrant, meaning that not every woman that has BRCA1 will go on to develop a breast tumor. Maybe 50 or 60% will develop, but that means 40% of women that would have that mutation in the germline do not develop a breast tumor or some ovarian tumor or whatever else they could be linked to. So that means these germline mutations are also secondary risk factors, not the primary risk factor. The primary risk factor in all major cancers is chronic damage to the oxidative phosphorylation coupled to substrate level phosphorylation. Now, what's interesting is what's preventing, okay, a woman inherits the BRCA1 or the BRCA2, and she doesn't ever get cancer. So why, even though she has that mutation, why did that mutation not cause breast cancer in that?
Yeah.
Because it never disrupted oxidative phosphorylation. Only the women that have disrupted oxidative phosphorylation go on to develop a breast cancer. Why did that woman who had that mutation didn't? The answer to that is we don't know. So it's called suppressive. We have known in genetics for years that there are suppressor genes. So that certain genes in another part of the cell or body can suppress the effect of one gene from doing what it's doing. So a primary cause of cancer is chronic damage to oxidative phosphorylation coupled to substrate level phosphorylation. We see that in all cancers, but the germline mutations may or may not do that. And we don't know why some women that would have the mutation or P53 mutations are the highest of all penetrants, about 90% penetrant, which means about 10% of people with the P53 don't develop cancer. And the answer is we don't know the suppressive effects of why another gene would suppress the effect of P53 for doing that. Now, getting back to somatic mutations, this is going to be very interesting. People say, oh, look at the P53 mutations in all of these different cancers, melanoma, breast cancer, all you got P53 mutations. Where did they come from? They came from ROS, R-O-S, spewing out of the mitochondria, caused the P53. So do you think those P53 mutations in breast cancer, somatic mutations, are causing the dysregulated cell growth? The answer is absolutely not. How do you know that? Because the new evidence is coming out showing, listen to this, normal tissues in women and other organs. We already find large numbers of driver P53 mutations in cells that are not dysregulated in their growth. It's proof that P53 it's not a primary cause of the cancer. So people are scratching their head to say, well, my God, we're loaded with cancer driver mutations, because the term driver came about as, only some mutations are the ones that cause dysregulated cell growth, and therefore we define them as drivers. Okay, but now we're finding driver mutations in P53 and other driver genes in normal cells that never become dysregulated. Mitochondria are perfectly healthy in these. They're perfectly healthy in those cells, therefore they're not going to get cancer unless your mitochondria are damaged. So clearly, we're now finding that all these mutations are secondary downstream effects of damage to oxidative phosphorylation. So as a result of this, we got 1,700 people a day dying from cancer because we're focusing on things that are not relevant to the problem. It's amazing. It's frightening. It's scary.
I want to stay on the more specific cancer types. I want to ask you, what about pediatric neuroblastoma? What can you tell us about the etiology and what about other, I know in your writing, or at least my understanding from your writing, is that cardiac and skeletal muscles can't survive without oxidative phosphorylation. I'm curious about the metabolic theory of cancer explaining soft tissue sarcomas like rhabdomyosarcoma in children, or bone sarcomas like Ewing sarcoma or osteosarcomas.
Yeah. So I looked into osteosarcomas and I looked into all these different kinds of cancers. And when you look at the mitochondria, they're defective. They have no cristae, broken cristae, and they're fermenting. So clearly they're all shifted to the same metabolic problem that you see. Now, childhood brain cancer, now we just developed a therapy that's under review right now that will be a blockbuster if the field will allow us to use it to manage childhood cancer. But we found that even kids that are like a few weeks old or even a couple of years old, these cancers are fermenters. They're throwing out lactic acid and succinic acid. So clearly they're fermenting. What happened to these kids? So my colleague here, Dr. Lindegren here in Boston College, has just shown that these rise in childhood cancers are the results of an infusion of all kinds of carcinogens in the utero situation. We have microplastics. We have all kinds of new, what they call forever chemicals that are now increasing in the environment. So any of these childhood developing brain or developing tissues that would be exposed to some of these kinds of chemicals could cause structural damage to the mitochondria, leading to compensatory fermentation. Because when we actually look at the solid tumor and see what they're doing, they're fermenting. They're not any different than adult cancers. The problem is they're happening at a much earlier stage in development, and we're seeing this. So clearly, why do we have childhood cancers? All we know is that the childhood cancer is fermenting. How did those mitochondria in that childhood cancer become damaged, leading to dysregulated cell growth and a fermentation metabolism? Obviously, our environment exposure to all of these forever chemicals, these various kinds of chemicals, are increasing and giving us more cancer. And we're seeing it, myself included. I see all these young people emailing me. They have cancer. They're in their late 20s, early 30s, and they're getting all these cancers. This used to be really predominantly in people over 50 and 60 years of age. Right. Now we're seeing colorectal cancer, all kinds of cancers. And I'm saying, what the hell? And it's our diet and lifestyle. We're in a more toxic environment. We're in a provocative diet and different lifestyles than we evolved to have. And cancer is just a consequence of our changing environment. It's not the result of our genes. It's the result of our exposure to chemicals and our diets and lifestyles.
I want to turn, okay, so that's very interesting. But I want to turn to cancer therapies under the metabolic theory. So how do the metabolic theories, such as the ketogenic diets and this caloric restriction, specifically target the cancer cell metabolism without adversely affecting the normal cells?
Yeah, well, this is how we evolved. We were always in a state of nutritional ketosis. Our Paleolithic ancestors of several 100,000 years ago lived in a very harsh environment where food was scarce. So we had to use tremendous amounts of energy to kill animals, chop them up, eat them, and there was very little carbohydrate. Maybe we could get some during seasonal ripeness of fruits or things like this, or some honey or something like this, but it wasn't abundant in our environment. So now we're in a very different environment, and we realize that when you're in a state of nutritional ketosis, where blood sugar is low and ketones are elevated, which is what we are evolved to be in, a state of we evolved in a state of nutritional ketosis, mainly because we didn't have access to highly processed carbohydrates in any significant way. In order we store fat because that's our lifeline to survival when there's no food. So we mobilize fat, and the fat is like a long chain of carbon hydrogens. It goes to the liver, and the liver chops it up like a wood chipper, and they come out with water-soluble ketone bodies, which the brain can burn readily. The brain can switch from glucose to ketone bodies when we don't have enough glucose in the bloodstream. So we were always in a state of ketosis. The heart can use ketone bodies, and I know the bioenergetics of a ketone body can give us more ATP efficiencies than can carbohydrates. Believe it or not, we're so much more efficient burning fats and ketones than we are ketones, especially not so much fatty acids. But our environment was that. Now we're in a very different environment, but we know the tumor cell needs the glucose to grow. So it's very hard to get cancer if your mitochondria are always in a state of nutritional ketosis. That's why cancer was extremely rare or non-existent in primitive tribes and our ancestors. So we didn't have cancer in the Paleolithic period. If we did, it would be maybe a lung cancer from sitting next to a fire all day long or something like this. But it wasn't a debilitating disease. Albert Schweitzer, the humanitarian physician who studied African tribes, was always shocked by how little cancer these Africans would have. They had parasite infections and other kinds of things, but cancer was extremely rare. Yet as soon as the Western diet and lifestyle entered into the society, all of a sudden you get chronic diseases, cancer, diabetes, and these other kinds of things. So what's happening now is that we're seeing more abundance of carbohydrates, which leads to greater inflammation, which leads to emotional distress. It leads to lack of exercise. And don't forget, our ancestors were very exercised. They exercised all the time. They didn't eat a lot of carbohydrates, and the cancer was extremely rare. So now we have a very different situation.
Given the heterogeneity of cancer, shouldn't the metabolic therapy be tailored to individual patients' metabolic profiles for optimal effectiveness?
Yes,
Right?
and that's what we do. So we just wrote a very detailed protocol. My colleague next door here, Dr. Thomas Seyfried from Czechoslovakia, he's an MD, PhD. He just, myself and some others, we put together this really comprehensive treatment protocol to manage cancer. And knowing that this problem exists, now we did it for glioblastoma because they have nothing. They haven't made any advance in glioblastoma in 100 years. Mm-hmm. So we developed this for the glioblastoma because they really can't say they have anything better. They have nothing. But if you look at the way we wrote the protocol, it'll be good for every kind of a cancer, whether it's a bone cancer, a blood cancer, a colon cancer, a breast cancer. They all have the same problem. They're driven by a fermentation metabolism. So this therapy would work and be effective in managing all kinds of cancer. But let me get back to your question. You're right. So people come into the clinic and they have all kinds of comorbidities besides having cancer. Some of them have parasitic infections. Some of them have type 2 diabetes. Some of them have hypertension. Some of them have high blood pressure. And besides, they have a tumor. So you have to take that person and bring them back to a better state of metabolic homeostasis. And the way we start that is we put people on a carbohydrate restricted diet, 0 carbohydrates for at least 10 or 14, 20 days, something like whatever is. And then we use the glucose ketone index calculator, which we developed here at Boston College. Couldn't patent it. They say you can't patent formula. So anyway, it was now put into the Keto Mojo meter. So you can buy it on Amazon. People can track their glucose ketone index just with a blood meter. You take a prick of blood, drop of blood, you can measure the blood sugar, and then you measure the blood ketone bodies. when the ratio comes down to 2.0 or below, meaning that the amount of sugar and ketone body, glucose and ketones are near unity, your cancer cells start getting hammered. Why? Because the brain and the muscles have first dibs on the available glucose, not the tumor. The tumor is like the lowest group of cells that have dibs on this. So they start to get restricted in their ability to grow fast because you're slowly lowering their key fuel and bringing the human body back into a state of homeostasis, increasing exercise, making sure your GKI gets lower and lower and lower into that special therapeutic zone of 2.0 and below. Now the tumor cells start to get hammered. They're not growing as fast because you're choking one of their two prime fuels, which is the glucose. And as we get into that low GKI, then we come in, we pulse. That's why I developed the press pulse therapeutic strategy based on paleobiology, where you can restrict chronically one fuel. We don't need glucose. We can switch to ketones and fatty acids. We don't need glucose. The tumor cell absolutely must have this glucose. So we slowly restrict one of the two fuels driving or maintaining viability of the tumor, and then we come in and target the glutamine. Now, glutamine has to be pulsed. You cannot chronically restrict glutamine because you're going to do damage and harm your gut, harm your immune system, you're going to cause physiological problems because both the tumor cell and the normal immune cell need the same fuel. So you can't be stupid about this. You got to know systems biology and systems physiology. You want to maintain a strong and healthy immune system to pick up the dead cancer cells. But if I chronically hit glutamine, I'm going to paralyze my immune system, leading to infectious clumps of tumor cells in the tissue. So again, this is press pulse therapeutics. So you take patients, and as you're right, many of them have very different metabolic states. And we have to bring those patients slowly back into a more homeostatic metabolic state and then come in with the glutamine targeting drugs, pulsing them and slaughtering these tumor cells because now they become totally vulnerable to this kind of strategy.
But when you have a patient that already has the tumor, and for example, you want to bring them into this zero glucose, zero carb.
Well, it's not zero glucose, it's low glucose.
Low glucose. But that's not easy, right?
Well, we evolved to do that. That's who we are. We're always in low glucose as our ancestors were.
Yeah, no, so I guess my question is more, is there enough time? Like, what is the time frame if you already have the tumor is growing and feeding on the glucose and the glutamine between bringing the patients down to a level that's low enough and then being able to do this pulse therapy that you're saying?
So usually when, not always, but majority of people, when they're first diagnosed with cancer, they're not always, some people say, I didn't even know. I went to the doctor. I had this little pain or I had a lump or something like this. Now he tells me I've got this cancer. Well, you're not going to be dead in 12 days from that lump, right? You have plenty of time to shrink that tumor down. And then when it's down, it's okay. It takes a couple of weeks. We're more concerned about glioblastoma because, you got a raging brain tumor. Is there a watchful waiting period where we can shrink down the tumor before the surgeon comes in and debulks the tumor? Surgery is an absolute crucial part of managing many cancers. So if I take that tumor that's first diagnosed, usually we always think it's better to diagnose it by non-invasive technology, either PET scan, MRI, CAT scan, or something like this. Poking the damn thing, you run the risk of spreading these tumor cells throughout your body by creating inflammation directly in the tumor itself, leading to a metastatic spread through a mechanism that I know all about as well, which I haven't even discussed yet, the origin of metastatic cancer, which we know all the metastatic cancers have characteristics of macrophages, which are part of our immune system, driven by glutamine and glucose. So I know how to manage metastatic cancer. So the problem here is that we have to shrink this tumor down to a point where it's not as angry or inflamed as it was. And then the surgeon and the oncologist will say, okay, I'm looking at something now not nearly as bad as it was. Let's go in and cut it out and see if we can cure the cancer, because the very obstacle, this tumor, is no longer as angry and inflamed as it once was. Now, that could lead to a cure right there. Now, if that doesn't work, the tumor becomes indolent because you're keeping the glucose down, and then you constantly pulse the glutamine. So it's a staged strategy. And, let's be honest, when during standard of care, if you're diagnosed with cancer, They go in there like a bull in a china shop. They start hacking you. If they do that, remove your breast, take out your bladder, or take out a part of your tissue. And then they start poisoning you, and then they'll radiate you. And the next thing you know, your hair's falling out. You feel terrible. Your microbiome is shot to hell. What are you doing? These people have no clue what the hell they're doing. They don't know it's a metabolic problem. We can get all of those things taken care of in a far less toxic way with an outcome that's far greater than what we're doing to these poor souls.
Are,
Um, yeah,
are there clinical trials, published studies that have been conducted to test the efficacy of the metabolic therapies in
no,
cancer?
no, of course not. Well, who's going to do it? They think cancer is a genetic disease. If you think cancer is a genetic disease, you're not going to be doing what I just told you to do.
But are you checking it in your clinic? And do you have
Well, I don't have
outcomes?
a clinic. I
Uh,
work with independent physicians who
right.
will email me and say, "Geez, we're starting to use your metabolic. It's unbelievable how these cancer." Well, they're not right. I
They're not
bust my
writing
ass
about
right.
it.
I write these case reports up. We've published papers on advanced breast cancer, advanced lung cancer, brain cancer. In small, oh, I don't believe you. You got an N of 1. I don't believe you. You got an N of 4. I don't believe. What the hell, man? If I had a drug that did what metabolic therapy does, I'd be the wealthiest guy in the world. They say, "Whoa, my God, look at what this guy, how much does it cost?" It doesn't cost anything, man. It can't make any money on it. It just works really, really well. But you have to have a knowledgeable patient and a knowledgeable oncologist or physician. When the patient and the physician are working together as a team, and the patient and the physician both know what I just said to you and have read the scientific literature and have a clear understanding of what these tumor cells need to survive, then you get success. We get tremendous success when the patient and the oncologist are knowledgeable.
Could these metabolic therapies be integrated with the existing conventional treatments, such as chemo and radiation? And could there potentially be synergistic effects as a result?
Absolutely. We showed that already in the paper we published out of our friend's clinic in Istanbul. We can lower chemotherapy drugs and the amount of radiation needed. We can lower the dosage, timing, and scheduling of these things to match the situation. So yes, we're finding that when the cancer is under metabolic attack and stress, they become extremely vulnerable to some of these chemotherapies. But you don't need to use as much as you would once use. You can get away with half the dosage, one time, one-third the dosage. So the collateral toxicity is reduced while therapeutic efficacy is enhanced. The problem is the oncologists are trained only to use this and don't deviate from this rigid procedure. They're not capable or permitted to do what I just said, reduce the dosages. We have all of the tools necessary to manage cancer effectively, non-toxically, we don't need to invent anything more. We have everything we need. We just need to know how to use better the tools that we already have. Even these immunotherapies that could cause hyperprogressive disease and off-target effects could become extremely vulnerable. If you take an angry tumor that has many different kinds of cells all fermenting and growing in a dysregulated way, and you shrink them down, they all have something in common because they survived this metabolic stress. Now you come in with an immunotherapy and you polish off the survivors. You don't go after the tumor at its strongest and most angry state with a therapy that might only kill a partial number of cells and risk the possibility of spreading the tumor to the rest of the body. So again, once you realize it's a mitochondrial metabolic disorder, the ways we approach the disease and manage patients will be very, very different. But we're not getting rid of everything. We're just learning how to use it in a more effective way. What's wrong with that?
Oh,
What is wrong with that? Nothing, right?
what? No, yeah. Uh, what would you say is the most pressing research question in the field of cancer that you'd like to see addressed by researchers?
Well, I think we need to recognize cancer as a mitochondrial metabolic disorder. I think we need to, because it's the theory under which you view the problem has to be the correct one. And I go back and I say these theories in science have had huge advances to mankind once we realized which is the correct theory to explain a phenomenon. For 1,800 years, the world thought that the Earth was the center of the solar system
Yeah.
and the sun and all the planets revolved around and many people still believe that. They still actually believe that the Earth is the center of the solar system. After 1800, all these years that we've Copernicus and Galileo and Kepler and all these guys, it was the geocentric theory where Earth was immovable and planets and suns circle around the Earth, and you got this big thread ball of mess going around. And then when you put the Earth, the sun in the center of the solar system, the Earth just became another planet circling around the sun. Everything became more clear. Advances in science, tremendous. Once we put mitochondrial metabolic theory in the center of the cancer problem, the advances are going to be enormous. And not only that, we're going to start managing diabetes, all these other chronic diseases, all related in one way or another to mitochondrial dysfunction. But right now, the field is not there. And when the National Cancer Institute says that cancer is a genetic disease, we think the people that are writing that must know what they're talking about. It's apparent they don't. Either they don't or they cannot. They might be yoked by a pharmaceutical system and a hospital system to keep the status quo. The status quo is extremely profitable. Everybody benefits except the cancer patient. So again, that's the power of the theory. The power of the theory. So I would say we need to invest more energy in substantiating. Let's compare and contrast the somatic mutation theory with the mitochondrial metabolic theory. I have already seen the hard data to support that cancer is a mitochondrial metabolic disease. That needs to be highlighted. More experiments must be done to target glucose and glutamine simultaneously while we're putting the patients in nutritional ketosis. That has to be perfected. I have just the outline and the framework and some clinical data, case report, clinical and preclinical, a lot of good preclinical data. So we have to target the two fuels together, not one at a time, not in a non-ketotic state. You put all that together in that package, and you will now begin to see management of this disease in a way so much more effective than has ever been done before. Will it work on everybody? No. Will it work on the majority of people? Yes. Okay, but we have to do the research, both preclinical, clinical, and scientific testing the theories. And once you do that, you will then move this entire field forward into knowing that this is a manageable disease. Is it going to be easy? No. Why? Because I'm going to have to give up certain things that I like. Now, of course, everybody would love the drug. Rather than going exercising and eating low carbohydrate foods to reduce weight, I can shoot up Ozempic and I don't have to, you know what I'm saying. Human beings always want the easy way out. Well, for cancer, there might be just this way. Or you can, listen, we're not going to stop anybody who likes high dose poisons and radiation. If they want that, they're always can have that. But we think there's a better way that most people would probably gravitate towards. The problem is they go to the oncologist, and the oncologist says, I never heard of any of this. Glucose has nothing to do with cancer. You hear it all the time. The patients come to me, my oncologist slapped me down, said all my stuff, it hasn't been proven by clinical trial. If you have enough common sense and understand biology, you should be able to see the connection. They don't look at the literature. They don't read the scientific literature. What are you going to do?
Very interesting. But we are coming to the hour and I need to close. And we have a tradition on this podcast where we ask our guests, that's you, to provide actionable advice for our listeners. So the question for you is, what feasible, immediate lifestyle or dietary recommendations would you suggest for individuals aiming to reduce their cancer risk based on metabolic principles? And would it apply to adults and children alike?
Well, yeah, certainly it's very hard to get cancer if your mitochondria remain in a healthy state. Nutritional ketosis protects the mitochondria against radical damage and enhances the energetic efficiency. So, which makes metabolic therapy both good for prevention, and if you do happen to have cancer, with glutamine targeting, an add-on, you can manage the disease. So this is one approach that can both be effective for prevention as well as treatment, which the current standards of care certainly cannot do that. So, but for children, we have a childhood obesity epidemic. That should not be. These folks need to know those little kids are at risk now for cancer, diabetes. Their lifespan is going to be shortened. They're not healthy in that, and they may look healthy and they may be able to run around, but that's not a healthy state. The kids should not be obese at 6 and 7 years of age. What is this? Is it lack of knowledge, parental neglect? I don't even know what you want to call it, but the education has to get out there to let the parents know this is not healthy. High processed carbohydrate foods on a regular basis is not healthy. Okay, it's no more than you would expect a lot of little kids to be smoking cigarettes. Nobody want to see go into a kindergarten classroom and all the little kids are smoking cigarettes. People would say, what are you guys crazy? But we have a lot of little obese kids in kindergarten, first grade. I said, what's going on with that? Should not be. Exercise needs to be exercise. Limited consumption of high processed carbohydrates. These are some of the preventions that you can do. Education. Okay, the Surgeon General just came out the other day and he said, consuming alcohol puts you at risk for cancer. And it's true. It does for certain kinds of cancer. He said colorectal cancer, liver cancer, and some of the breast cancer for women. But he didn't say anything about the big dog, which is the obesity epidemic. That puts you at a hell of a lot risk than these other things, not that the other things don't. So why don't we get to the heart of the problem? Because it's controlled by special interest groups, not to have this information disseminated among the population. God forbid everybody gets healthy. What are we going to do? Everybody looks like a Paleolithic man running. Everybody's lean and mean. But we're never going to get there. But at least we know what we can do. And that's why we developed the glucose index, because a lot of people without cancer can get into a lower GKI just to stay healthy. So that's one thing. It's quantitative. Now we see a lot of young people, they can have an app on their phone. They get a GKI value. It immediately goes to their app. So you're in constant awareness of your own physiological state. I think that's going to be exciting. So some of the technologies coming down the pike will be able to let people know what they should or should not do, or at least guidelines. And I'm not here to say everybody should stop eating these kinds of foods, because I enjoy them too. I love a big hamburger. Let me be honest with you. I mean, all that stuff, but I don't do it every day. I try to I do exercise, walk 5 miles a day or whatever. The idea, we're all going to die from second law of thermodynamics. We just want to prevent it as long as we can.
I think education is key. But on that note, I thank you very much for joining us today. And thank you to all our listeners for tuning in, and we'll see you next time. As we close, I want to thank you for joining me on the Her Notebook podcast. I hope today's conversation brought you some knowledge and sparked new insights. If you enjoyed this episode, I humbly ask that you please remember to click the subscribe button below. Until next time, keep flipping your notebook pages and embrace the journey to a healthier, happier you. See you next week.